Good morning, students. So yesterday we discussed about the structure of gene, right? So what are the parts of structure of gene? What are the parts? So we discussed various parts. What are those in the structure of gene? Promoter region, then we have coding region and introns and extrons, right? Exons. So what is the role of introns? So keep in mind that this is the gene. So we have introns and we have exons. Exons will code the proteins, right? So previously I said that there is no function of introns. Introns not going to code any proteins, right? But introns will act as a regulators to the genes. Introns will act as a regulators of the genes. Okay. The length of the intron, what gene when need to be coded, the timing of the gene coding will be determined by intron lengths. So introns acting like a molecular clocks for regulation of the gene activity or to code the genes. Okay. And yesterday I said in the promoter region we have minus 35 region and 10 region. So there are some regions, these are called promoters, and you have the coding region and you have the last of final introns and exons. So that is what we learned. Then today we will go into role of introns. Okay. Then role of introns and exons. So let me write here. Role of introns and exons. I'm writing intron here. Then exons. Then we have prokaryotes and we have eukaryotes. Okay. Now listen. One interesting thing I need to discuss first. Introns are absent in prokaryotes, whereas introns present in eukaryotes, exons present both. In case of exons in eukaryotes, multiple exons are present. Multiple exons present. It has one significance. A same gene here, see, a single gene will code one protein. In case of prokaryotes, one gene will code only one gene, one protein. Okay. But in case of eukaryotes, the same gene can able to produce multiple proteins, protein 1, protein 2 and protein 3. So this is one interesting phenomenon in eukaryotic genes. Okay, this phenomenon is exhibited by one type of gene called as tropomyosin gene. One example, okay, tropomyosin gene. So this tropomyosin gene can able to code multiple types of variety of proteins from a single gene. Okay, yeah, that is it. Such genes are called as isoforms. Such proteins are called as isoforms of a single gene. Okay, isoforms of this particular gene. So same gene will code multiple types of proteins. So that is one thing. Then next one we need to discuss is exon shuffling. Exon shuffling. Exon shuffling means this exon shuffling only present in eukaryotes where events including new genetics and new novel combinations. Okay. How to determine exon shuffling? See, I need to add one more point. I will write here gene 1, gene 2. This is intron, this is exon, this is intron, this is again exon, gene 3. So in eukaryotes, what's happening? Splicing. There is an activity called splicing. What is the meaning of splicing? Splicing means it will cut all the introns and it will cut all the genes, and all genes will be restitched together. Gene 1, gene 2, gene 3. Okay. This restitching of genes is only present in the eukaryotes, and the removal of introns is called as splicing activity. Okay. Now, upon stitching together the genes, sometimes in eukaryotes, these genes can undergo shuffling also. Okay. Shuffling leads to variations in the variations within the body. So, if our parent, mother was somewhat dark skinned, father was fair skinned, and baby has a chance of getting a 50-50 chance, dark or fair skin, because there the shuffling of genes happening of both paternal and maternal, father and mothers. Okay, those variations are happening because of shuffling of genes after splicing. So this gene shuffling only present in the eukaryotes but not in the prokaryotes. Prokaryotes straightforward, one gene, one protein. Okay, and also introns will, as I said, it will go for regulation. So this is all about introns and exons. Then let us go to transcription. Next one, very basics only again. Transcription also we started. There is a difference between transcription in the prokaryote and eukaryote. So now I will discuss about transcription. Actually, what I am feeling was we are still in the loop of few topics. We need to cover many topics actually. Anyway, see?
Sir,
yes.
डिस्कसिंग होने के बाद ये इंट्रॉन निकलता है तो एक्सॉन्स जुड़ते हैं ना, तो उस मेथड को क्या बोलते हैं? लाइगेशन. तो यहाँ पे सफरिंग तो नहीं होगा फिर क्योंकि सेम आएगा ना?
Yes. No, obviously after restitching it will be same order, but there is a phenomenon where they can also undergo shuffling. So there is an alternative mechanism of shuffling also. Maybe eukaryotes are encouraging the shuffling to get variation among them. So there is a different mechanism. We don't need much. That will be biotechnology. Okay. Next one. Transcription, we already stated there is an initiation, elongation and termination, and I have discussed the initiation, Tata box in eukaryotes, whereas AT, AAT and GC, that is for prokaryotes. So already transcription finished, right? Do I need to recheck, this one? So a quick architecture of genes, transcription. So let us go to transcription. So in every transcription there are three phases as we discussed. What are those three phases? Initiation, elongation and termination. So how the initiation happens in prokaryotes, how the initiation happens in the eukaryotes, then how the elongation happens in eupro. That's it, okay. Already we finished it. In case of termination in eukaryotes, there is a poly A tail, whereas in the prokaryotes we have row dependent and row independent. This we cleared in the yesterday's lecture. So I'm just keeping this transcription also, it is also over. Next one will be translation. So what is the, so before translation there is one more concept. After transcription we have post-transcriptional modifications. Then we have translation. What is the overall goal of doing transcription? We have a DNA. DNA converts to mRNA. Right? So now this mRNA will be sent outside the nucleus, and it will be transferred to the nearby ribosome, where the translation happens. Right? This will be taken care by the tRNA. tRNA will take the mRNA. It will guide towards the ribosome, transfer RNA. That is the role of transfer RNA. Now, what they are saying is, see, when we are sending a pure RNA into the cytoplasm, Okay, from the nucleus, when we send pure RNA to the cytoplasm, there is a problem. The problem is there is a chance of losing of some, the terminal ends of the mRNA might lose due to the cytoplasmic isozymes activity. In the cytoplasm, there are some enzymes that will destroy the proteins. Now, these enzymes can mistakenly damage the mRNA. How to protect mRNA during its journey towards the cytoplasm? So what we did was, we are going to do post-transcriptional modification to the mRNA. So we are trying to protect the peaks of the both mRNA. So by protecting it, we are able to maintain the stability of the mRNA while it's going towards the cytoplasm, towards the journey towards ribosome. Okay. So such activity is called as post-transcriptional activity. So there are multiple approaches of protecting the mRNA. So those approaches we will see here. Multiple there. Actually, phosphorylation, methylation. I will see here. Post-transcriptional modifications. I will just do modifications. This one, PPMs, post-transcriptional modifications. See this diagram? So there are multiple approaches actually. In a nutshell, we have methylation, phosphorylation, acetylation, cleavage, complicated, multiple pathways there for protecting the mRNA. So just remember this one. You need not to go much, just try to understand the basics. So how the mRNA was protected post-transcription. Okay. So there are some approaches: methylation, phosphorylation, acetylation. Okay. So there are few methods of protecting the mRNA upon transcripting into transcription. Yeah. So this is about post-transcriptional modifications. So obviously, when you are adding some phosphate groups, there is an enzyme called phosphatase. When you are adding some methyl group, then there is an enzyme called methyl transferase. Whatever modification we are doing, the appropriate enzyme involved there. So that is about modifications. Then splicing also done. Alright. Then coming to the translation. Now listen. In the translation, what happens? The gene code will be converted to a protein, right? So this translation happens in triplet codon. That I want to discuss. Triplet codon. Right. Triplet codon. Where is it? This one. So this is it. So we can say this is a periodic table for life. So we have total 20 important amino acids. All these 20 amino acids will code almost all the proteins present on planet Earth. Okay, all life on planet Earth has only these 20 proteins. Right? What is the significance of proteins? Proteins will give the structure. So all life need one type of structure. When we see the external morphology, not only external, but also enzymes, everything has a structure. So we have a universal dictionary for this coding. Okay. So any three base pairs of the DNA will code one protein. Okay. So one amino acid. To be precise, sorry. Okay, so we have 20 amino acids, and this is the sequence and their appropriate amino acids. Okay. Now, in this we have two important things. One was stopping codons. Stopping means once we get UAA, UAG or UGA, it will stop the translation. Okay. And we have initiation also. Initiation is promoted by Meteorology. Okay. So AUG will initiate the translation, whereas these three will terminate the translation. And in between the base pairs will be the other amino acids. So this is how it will stitch amino acids one by one to make a protein. So in translation, what happens? This tRNA will submit the mRNA to the ribosome. And what is a ribosome? Ribosome is also a type of RNA actually. Ribosomal RNA. Okay. Ribosome will, it will take any three codons, A, U, U. What is the amino acid of AU? Check there. Is that AUU? See, AUU is LLE means isoleucine. Isoleucine. Okay. So any three codons, if you take them, it will make one amino acid. So one amino acid. Next to three codons, another amino acid. So a strand of amino acid is called as a peptide. So a multiple peptides will make a polypeptide. Okay, polypeptide. A group of polypeptides will make a protein. Right. A complete, tertiary, quaternary. So those structures. Fine. So this is the translation. Just you need to know this table. And yeah, we have the codons. So let us see the simple steps in translation. So translation means it is a process of synthesizing proteins from mRNA template. This occurs in the ribosome, where codons in mRNA are decoded into specific amino acid sequence. So the phases of translation, again we have initiation, elongation and termination. Initiation will start with the starting codon, that is AUG, and initiator will be tRNA, obviously. Then elongation. So the ribosome moves along the mRNA, reading the codons. Oh, it's not mRNA moving through a ribosome. Ribosome itself moving along with the mRNA, right? So then tRNA binds with specific corresponding amino acids to the stitch. Then termination. That's it. You have three terminating phrases: UAA, UAG, UG. That will terminate the translation. That's it. So the role of mRNA provides the codon sequence for protein synthesis. tRNA will transfer specific amino acids to the ribosome. Then ribosome, it will catalyze or it will do the translational activity. Codon, any three base pairs will make one codon, that is called triplet codons. And anticodon, complementary triplet on tRNA. Okay, base with the mRNA. Obviously, complementary. What do you mean by complementary? When this is coding, AUU is coding. In the tRNA, what base pairs will be there? Complementary base pairs will present. G. Okay. Keep in mind, uracil was replaced in RNA. Okay. So that's it. So this is all about the translation. Then coming to the types of ribosome, this number is important. Okay. In case of prokaryotes, we have 70S ribosomes, that include 50S and 30S. Whereas in eukaryotes, we have 80S ribosomes, 60-40. Just to remember, okay. So we have two types of ribosomes, both in pro and eu. In case of pro, we have 70S, that subdivided into 50-30. And in eukaryotes, we have 80S, that subdivided into 60 and 40. It's not representing the sum of any two, it's just the number, keep in mind, okay? Yeah, so that's it. So this is all about the translation. And as I said, multiple genes, same gene can translate multiple types of proteins. That is present only in eukaryotes but not in prokaryotes. Prokaryotes, one gene, one protein. whereas in eukaryotes, one gene can code multiple types of proteins. So that is the uniqueness in eukaryotes. Then this is the gene code. What are the 3 base pairs and the corresponding amino acid. You need not to buy that, just have one idea. That's it. So that's it. Then genetic code organization already done. Same things. Let me cross check your syllabus once. So in the syllabus, we have DNA as a genetic material that we completed, then flow of genetic information, Griffith, Avery and Eschrich's experiments. Then we discussed about structure and application of DNA. Then we discussed about the structure of gene. Then transcription we studied. There are 3 phases of transcription for both transcription and translation. And the other one is genetic code. Genetic code means this one, or this code, this table. Okay. So what is the 3 base pair genetic code and the corresponding amino acids? Okay. The book of genetic code. Then organization of prokaryotic and eukaryotic genome. This is the final topic in chapter 1.1. Organization of prokaryotic and eukaryotic genome. Yeah, let us see that. So how organization of prokaryotic and eukaryotic genome. So genetic code we already know this one. Triplet, degeneration, start and stop products done. Then significance of genetic code, of course protein synthesis, evolution, gene regulation and biotechnological applications. Then organization of prokaryotic. Let me go directly to prokaryotes. Actually, I'm expecting a diagram where it will show the hierarchy of a genome, which is not available in these notes. Organization. Okay. This table I want. Yeah, simple. Keep in mind, in prokaryotes, the DNA structure is in circular form, whereas in eukaryotes it is in linear or multi-chromosomal form. So chromosomes present in the eukaryotes, whereas in prokaryotes it is in circular, open. It is not enclosed within the nucleus. Then chromatin. Chromatin is absent in the prokaryote, whereas it is present in the what is the significance of chromatin? See, this is the DNA. Now what happens was, in case of bacteria, the DNA was circular DNA. That's it. This is bacterial DNA. Whereas in eukaryotes, now what it did was, it compressed the gene. It compressed it. It's in linear form. Now the genes are compressed. That compression is facilitated by some proteins. You can imagine we compress a spring and we kept some bolt in between the spring to lock the spring tightly. Those bolts are called as histone proteins. Okay, histone proteins. Due to this tight compression of gene, it will take a color, a dye, that is called chromatin. Okay, since it's uptaking this dye, that is called as chromatin. So this is what they are saying. Chromatin absent in the prokaryotes, whereas chromatin present in the eukaryotes. The chromatin was present and associated with the histones. Histone proteins, what is the function of histones? They will tightly pack the gene into a compact structure. Okay, so that is the role of histone. Then location of the genome. So present in the cytoplasm in case of prokaryotes, whereas the genome was present within the nucleus in eukaryotes. Then next one, gene organization. Organons. Okay, operons is a common term for both introns and exons. There is no specific introns within the bacteria, but it has a type of thing called operons. Okay. Whereas in case of eukaryotes, we have monocystonic genes with introns. Simply introns are present in eukaryotes, introns are absent in prokaryotes. Introns are replaced by different thing called as operons. Okay. Next, replication origins. Single origin of replication, multiple origins. Yeah. In the same DNA, multiple replication happens simultaneously. Replication fork will be opened in multiple sites of the same gene, whereas that's not possible in the prokaryotes. Clear? So, if I ask a question about organization of a genome in U and Pro, you can elaborate same points. If you want, you can go depth by showing some diagrams. Depends on the type of question, 2, 5 or 10 marks. Right? Yes. Yeah. Then, what is this plasmids? How plasmid DNA is different from circular DNA? Any idea? You already stated it. This is only present in the prokaryotes, by the way. What is the role of plasmids? Have you heard about plasmids? Yeah, what are those? Yeah, they are extra DNA circulating present within the cytoplasm. Can we say cytoplasm for a prokaryote? It has cytoplasm, right? Even prokaryotes. So, yeah, we have a genome circular DNA. Apart from that, we have another circular DNA. Okay, the small pieces of DNA called plasmids. What is the role of plasmids? The Griffiths experiment you need to recall. What is the role of plasmid? Yeah. Yes. Very good. They will carry antibiotic resistance. They will carry, we can say that plasmids are like an immune system to the bacteria. Okay. So they will develop resistance to some drugs, and these drug resistant proteins will be transferred from one bacteria to another bacteria. Right. So plasmids will act as a vehicles for maintaining their genome, their resistance, drug resistance or any anti-bacteriophage resistance. Since we are discussing plasmids, I want to discuss one interesting thing. So what happened was, there is two scientists, Christopher Dorner and Emmanuel Charpentier in 2001. So they have found very interesting phenomenon in the bacterial genome. They want to understand how. So this is a bacteria and this is a virus attacking the bacteria. This virus is called as bacteriophage. Okay, but somehow bacteria able to recognize the specific mosaic virus. Let us take it is a mosaic virus. Okay, tobacco mosaic virus. Bacteria are very specific in killing those viruses. How bacteria evolved this immune system? It is a single cell living organism, but how, in what mechanisms it is identifying a specific virus? Got my point? Plasmids is different story. Plasmids are, some mutations happen in their genome and they develop drug resistance, and this drug resistance will be transferred horizontally or vertically from dead being to another being. That is a different story, plasmids. But how does a bacteria recognizing its enemy? How it's recognizing its prey? Lytic or lysogenic? Okay. Say lytic or lysogenic is a non-specific enzyme. Killing any proteins coming, foreign proteins coming means they will destroy the foreign proteins. But I am saying specific to the virus. How is it specific to the virus develop? Specific cuts. Yeah, very good. See, what these two scientists discovered was groundbreaking actually. What they discovered is, they saw some short palindrome means palindrome. What is the meaning of palindrome? Do you know? Can you give one example of palindrome? One, if you from right to left or left to right, it will be same. That is called palindrome in English. Can you give one example of palindrome? One palindrome word. Anyone? I. You can read EYEI or reverse also I, right? So similarly, in the bacteria they found AUGUGG. They found a specific palindromes within the genome of the bacteria, and they are properly interspaced. These palindromes are present in every proper same spacing. So they are confused. Why this bacteria having some? And by the way, these palindromes don't belong to the bacterial actual genome. Some foreign genome. Why bacteria maintaining some foreign genome in their gene bank? What is the use to the bacteria of maintaining some foreign genome? They didn't understood. They termed these genes as clustered. Clustered means they are clusterly arranged, regularly interspaced, short palindromic repeats. Palindrome. The word palindrome, repeated palindromes, clustered regularly interspaced, repeated palindromes present within the genome of a bacteria. So they say it has a CRISPR genes. Then what is the role of CRISPR? I will discuss. Then bacteria has a specific enzyme called CRISPR associated caspase, CAS enzyme. Caspase 39. Okay. CRISPR associated system 39. There is an enzyme simply. This enzyme is like a scissor. Caspase 9. Actually, there are multiple members. Anyway, caspase 9. Cas9. Okay. Oh, you studied about CRISPR there. It's very interesting. So, see, now bacteria has a scissor and it has a palindrome. So, what exactly happening was this scissor moving across the gene this is only cut at these palindromes. So whenever, by the way, these palindromes are the palindromes of the attacking virus. Simple. The bacteria evolved a immune system where it is maintaining its predator's gene. Maybe if I want to kill adult, genetically speaking, for example, I will maintain the adult's gene within my genome. So I trained my gene to recognize the foreign gene specific towards the adult. And whenever adult try to enter into my body, genetically speaking again, a virus, now my scissors will cut her gene. Because my scissors are guided via my gene pool, where the scissors are able to identify itself from adult's genes. Okay. So that's it. It's a simple mechanism in the virus, but it took almost 70 years to identify CRISPR genes in the bacteria. So what is the use of this CRISPR system? That is the next question. So, what they did? If a bacteria has a precise gene cutting scissor, this is one greatest tool. Because previously we want to do some hybrid techniques. I took a tomato which was spoiling within 2 days, and I know that there is one gene that will protect the tomato. I want to cut this gene and I need to paste this gene in the tomato. But the cutting and pasting is not easy. The molecular scissors, whatever we have, zinc finger nucleases or UIAHS, all these things are not so precise. We are getting 30 to 40% errors when we are cutting the genes. Then, can we guide Cas9 scissor to precisely cut a gene? Is it possible? Can we reverse engineer, or can we guide this protein to cut any known genes? So what we did? We exploited this system. We took the Cas enzymes. Then what we did? Very interesting. Within our nucleus, within our nuclear genome, first we need to introduce the target scissoring gene. Maybe I want to remove the insulin gene from my gene. He is a diabetic patient. He has a bad insulin gene. So what I did, I introduced a Cas enzyme and I introduced an RNA of the insulin gene. Now this Cas enzyme will guide through the genome and it will do the cut wherever we need, and that will be replaced by good gene. By the way, our DNA has a very good mechanism. If you cut the gene, it will replace with better gene. Okay? Yeah, where we studied the opposite. It will do proofreading, or something is missing here and it will replace it. See, today we can genetic engineering is possible. This is a genetic predisposed problem for us. Insulin gene we got from our parents. Today we can remove that insulin gene and we can replace it with a better gene.
So better gene के लिए हमको inject करना पड़ेगा gene of interest.
Gene of interest, yeah, we can get from any healthy individuals, and the gene of interest will be guiding the caspase enzyme, and that will bind it to it, bind the thing. So I hope you understood what happened. So some of the groundbreaking discoveries in CRISPR are, there is a person, I don't know from China or where, so by default he is a blind, by birth he is a blind person. The problem is, in his eyes he has rods but cones are not producing. The code, the rod and cones, right? Cones genes are absent in this person. So what they did, they reintroduced cone coding genes into the eyes. Simply, they kept a Cas9 enzyme along with these RNAs. And you know what happened? After 6 months of treatment, the person, the eye started developing its own cones. And this person has seen the world first time. A genetically blind person saw the world. That is really, first time we became gods. That is only possible by God actually, right? Yeah, we exploited the genes. When we understand the genes, we can do it. But still, CRISPR has one disadvantage. The disadvantage is, it is 99.1% accurate. 0.9% mistakes doing by the CRISPR. And that's a very concerning thing. You know why it's concerning? We need 100% accuracy. We can't touch CRISPR in the germline genome editing. Internationally that is banned. Why we are not going to touch germline gene editing? What is the reason? So maybe I will say one more thing. In the future we can make designer babies. We can design our own babies, whatever we need. Or my baby need a blue eyes, whatever traits we need. We can give those genes and we can make our, the specific characteristic traits in the babies. Now, what if you induce some mutations? That is a problem. The baby got a mutation and the entire baby's life is at risk because of our selfish desire. We had some design and we did it and we got a mistake and the baby is still alive till its death. Now the second problem, if the baby going for next generation, if it's going to reproduce, the mutation will carry on. Now it becomes an uncontrolled, something that nature hasn't seen before. Something that takes 200 to 300 million years to happen, such mutations going to happen in this immediate generation. So there are some problems with CRISPR still. So germline editing is not recommended so far, but in China, this idiot one person, he was internationally banned now. He took sperm and ovum from two parents. What is his excuse is, both of them are HIV patients. So definitely HIV patients will get a HIV baby. So what he said? He said that he will remove that CXCR5 gene, CXCR5 protein gene from the parents. So HIV needs this gene to infect a cell. So he said that he will remove by CRISPR. He removed them and he went for procedure IVF, and IVF gave two twins actually. And unfortunately, both the twins has 30 mutations in their body. They have joint issues. And they kept their life in a confidential state. So the mistake happened. That should not happen, right? Actually. Anyway, that is about the CRISPR. One more application. Actually, I like this CRISPR a lot. One more application, very interesting. See, we can eliminate the malaria forever from planet Earth. Very easy. You know, rather than killing the mosquitoes, let us genetically engineer the mosquitoes where they, we will remove the gene that is responsible for falciparum entry into the mosquito. So, if we re-engineer that and we allow the mosquito to go wild, there is something called a drive, gene drive, where this modified mosquitoes will be dominating the population. Okay, gene drive concept is there. So still we are maintaining the ecology, we are maintaining the ecosystem. We are not disturbing, we are not removing the mosquitoes, but we have removed the parasite within the mosquito by gene modifying. What do you think about forever malaria eradication by CRISPR? Do we need to proceed or not? We just take some 100 mosquitoes to release in the wild, and they will take care to eliminate the malaria. The still questions are, so today we remove malaria from the system, from the ecology. Is something new parasite coming that is resistant to this, it will definitely disturb the balance with the nature. And if something become very threatening to humanity that we never say because we did it first time, and it will go out of control. At least COVID is a manageable virus. It's not so threatening, life-threatening. Just imagine if it is a Ebola pandemic, game over. Ebola will cause blood clots. Yeah, Ebola is very dangerous. If Ebola is a global pandemic, definitely, all these institutes might be abolished by now. Half of the humanity wiped out. Anyway, so these are the potential applications of molecular biology. So because of molecular biology, we have developed vaccine in a matter of 6 months, 6 to 7 months. So yeah, that's the greatest achievement. Fine. So from plasmids I came up to here. Plasmids and genome organization in both prokaryotes and eukaryotes. See, I got CRISPR here somewhere. CRISPR-Cas9 in its specific genome regions. So yeah. So these are few applications of studying genomes. Yeah. Next one will be chromosome, that is karyotyping. Very interesting. I will discuss in tomorrow's lecture. Or shall I take lecture in your practical hour? 1 hour? Can I? We'll see. She's smiling. Okay. We'll see. Alright. Thank you, class. I given you the complete study material. Have you seen it? Almost 300 pages. It has everything. It has everything including 2, 5 and 10. You understood, right? Yeah. 21, 20, 25. Yes, your attendance class again. Mayam, Kanau, Dorcas. Who is Kanau? Oh, okay, fine. Dorcas. I haven't seen Dorcas so far. Vinayak means then Vinayak means you are the one. Okay. Then Zayed. Yes, sir. Zayed is the only student who came regularly. Zayed, Neelakshi, Priya, and this Morpella and Ashley came regularly. Of course, Aliosis also. Priya, then Garima. Yes, sir. Then Aliosis. Sandeep. Yes, sir. Morpello. Ashley. Only this Dorcas. Is she regular student? He or she? Dorcas. She. So, not a regular, right? Even if you don't know, then maybe register, but thank you.